Hello and welcome to part 2 of the chapter 19 lecture. Uh, as you recall, in part 1 of the lecture, we got a good look at a lot of different types of viruses and a lot of different ways in which they can reproduce and things. And so basically now we're going to take a few minutes and look at the second half of the chapter and finish up and see a lot more interesting things about viruses. So the first thing we're going to look at are the replicative cycles of animal viruses. And there are basically just two key variables that are used, remember, to classify viruses that affect animals. Whether it's got an RNA or DNA genome, and whether the RNA and DNA are single-stranded or double-stranded. And then the presence or absence of a membranous envelope. So RNA, DNA, double-stranded or single-stranded, and whether or not there is a membranous envelope on the very outside layer of the virus. So whereas a few bacteriophages have an envelope or an RNA genome, many animal viruses have both, as we'll see in a minute, and over the course of the next few slides that we'll look at in this chapter and in this lecture. So the envelope, a lot of viruses that affect animals have a membranous envelope, it's called. And so viral glycoproteins on the envelope bind to specific receptor molecules on the surface of the host cells in order to recognize which host cells they should try to invade and infect. Now, a glycoprotein, you probably remember glycoprotein from when you took General Biology 1. What does glyco mean? Do you remember what glyco means? For example, glycolysis. What does glycolysis deal with? Well, glyco actually means sugar. And so glycoproteins are basically proteins that have a sugar or a carbohydrate chain that's attached to them. Kind of usually large, complex proteins with some sort of carbohydrate that is sugar chains attached to them. And so this viral envelope is usually derived from the host cell's membrane. Derived from means modified from. So basically, in other words, the virus takes part of the cell's plasma membrane and it derives or modifies something from it to make the virus's own envelope. The envelope is basically the very outer layer of the virus. So as the viral capsids exit the host cell during lysis, they take some of the cell's plasma membrane along with them, and they modify them and make the viral envelope from that. Basically, here you can see you've got a virus, and it enters the host cell, and then it's got an RNA genome, and it uses that as a template to produce messenger RNA. The messenger RNA produces more capsid proteins, and they get glycoproteins from the endoplasmic reticulum. That's ER there, endoplasmic reticulum of the cell, and also the template is also used to make more RNA genomes for the new virus particles. So as the virus particles leave the cell, they take a little bit of, they basically coat themselves with a little bit of the plasma membrane with the glycoproteins on the surface, and that surface membrane then is the viral envelope, then the outer layer of the virus. And other viral membranes that might be present are actually made from the host's nuclear envelope, which is the membrane that surrounds the nucleus of the host cell. And they're then replaced, they might be replaced by an envelope made from Golgi apparatus membrane. So, they're making use of a lot of the different membranes inside the host cell sometimes. And the broadest variety of RNA genomes is found in viruses that infect animals. For example, retroviruses. Now, since they're called retroviruses, these are not necessarily viruses from way back in the past. Retro here actually doesn't mean back in the past. What retro usually means is backwards. For example, retrograde movement would be movement, backing up, like backing up a vehicle or something. So retro basically means backwards or reverse. So retroviruses are called retroviruses because they have something called a reverse transcriptase. You remember transcriptase? Basically, transcriptase enzymes cause transcription. And remember, back to General Biology 1, transcription is the process of using DNA as a template to produce RNA. But here, so transcriptase, the normal transcriptase, uses DNA to make RNA, but this reverse transcriptase from these retroviruses uses RNA to actually make DNA. So these viruses transform their RNA genome into a DNA genome by using their RNA genome as a template to make the new DNA molecule. And the reverse transcriptase enzymes do that. So, for example, the HIV virus, human immunodeficiency virus, is actually a retrovirus. And of course, it causes AIDS. So the viral DNA that is actually integrated into the host genome, during the lyogenic cycle, remember, is called a provirus. But unlike a prophage that infects plant cells, a provirus remains a permanent resident of the host cell DNA. And so when RNA polymerase is doing transcription, then the proviral DNA is transcribed as well into RNA molecules. And the RNA molecules function as both the messenger RNA to make proteins, and also function as genomes for the new virus particles that are released from the cell then. And here are a number of different types of viruses here. So there are a lot of double-stranded DNA viruses, the herpes virus, the polyoma virus, adenoviruses you may have heard of, the papilloma virus. The papilloma virus is the virus that they're testing for when you get a pap smear. The reason it's called a pap smear is because they're looking for papilloma viruses then. So there's a lot of double-stranded DNA viruses, a lot of single-stranded DNA viruses, and then some double-stranded RNA viruses, or dsRNA, or single-stranded RNA viruses. And then some single-stranded RNA viruses actually serve as a template for making messenger RNA. Or the retroviruses, as we've seen, use their single-stranded RNA genome as a template to make DNA then. So flaviviruses, and flavi means yellow, and so things like yellow fever virus, but also the Zika virus, you probably remember from the news a few years ago, dengue virus, West Nile, hepatitis C. So a lot of well-known and really dangerous viruses are flaviviruses that are single-stranded RNA viruses. And picornavirus then the rhinoviruses. Rhino means nose, so common cold. Also poliovirus, hepatitis A, and then the coronaviruses. So the coronaviruses are single-stranded RNA so SARS and MERS, and of course COVID. Here's some less known, paramyxovirus, filovirus, etc. So many different types of viruses that, and so here's another virus then. It's got a viral envelope around it, and there are glycoproteins on the envelope. And this is a retrovirus because you can see it's got reverse transcriptase. And so the capsid then releases its reverse transcriptase and its genome into the host cell. So basically the viral RNA is there, and it's got a reverse transcriptase. So the reverse transcriptase then produces a new strand of DNA there. And basically at first it's an RNA DNA hybrid, because the new DNA is produced using the original RNA genome from the virus as a template to make the new strand of DNA. Then they convert it to double-stranded DNA that goes into the nucleus and becomes part of the regular genome of the host cell as a provirus in the genome of the host cell. And then when that's replicated, then the new RNA genome is used here to partly as messenger RNA to make new proteins and reverse transcriptase enzymes for the new virus particles that are formed. And then the new RNA genomes that are placed into the new virus particles, and the virus particles are released then from the cell, complete with an outer viral envelope on it, also including glycoproteins on its surface as well. And you can see pictures of this, an HIV virus then in coming up to a white blood cell and getting into the white blood cell. And then also here's HIV leaving the cell, HIV virus particle coming up to the membrane, pushing out into the membrane, and then being released from the cell. So really great electron microscope photographs then. So these processes actually happen. We've got real photographic evidence that these types of things actually do happen, and the writers of the textbook are not just making these things up to sell textbooks and make the chapter sound interesting. And so here you can watch on your own time. There's an animation of the process of HIV virus reproduction cycle. So now we mentioned before then, viruses do not fit our definition of living organisms because they can't reproduce on their own. They can't even carry on metabolism on their own. So since they can only replicate when they're inside cells, those who are coming up with hypotheses about evolution, they think that maybe they evolved as bits of cellular nucleic acids that began to act independently on their own. And candidates for the source of viral genomes might be plasmids that are produced by bacteria, if you remember that from General Biology 1, or maybe transposons, again, something that was discussed in Biology 1. So these transposons, remember, are small sequences of DNA that can actually get up and move. They're small sequences of DNA that are normal parts of a eukaryotic cell, not a virus, but eukaryotic cell, and they normally get up and move from one place to another, maybe even move to a new chromosome. And so the plasmids and transposons and viruses are all mobile genetic elements, and so they're thinking maybe the original viruses were actually either plasmids and or transposons. then, and acquired new characteristics and some new abilities. The largest virus yet is the size of a small bacterium, and it's got genes then that encode proteins that are involved in translation, DNA repair, protein folding, and even polysaccharide synthesis. So there's controversies about whether this virus evolved either before or after cells, even. So viruses and then prions then. Diseases caused by viral infections affect humans, agricultural crops, and livestock, etc., wild animals as well. But there are also smaller things that are smaller than viruses. Smaller, less complex entities called prions also cause disease in plants and animals. So viruses could damage or kill cells by causing the release of hydrolytic enzymes from the lysosomes inside the host cells. Some viruses cause infected cells to produce toxins that lead to disease symptoms and cause disease in the animal's system and body. Other viruses then have molecular components like envelope proteins that are also toxic to the host animal. So a vaccine, we pretty much all know what a vaccine is then. Basically harmless, it's made up of harmless derivatives of pathogenic microbes that actually stimulate the immune system to mount defenses against that type of pathogen. And remember, a pathogen is some sort of thing, usually like a virus or bacteria, but a pathogen is just something that causes disease. So vaccines can prevent certain viral illnesses by stimulating the immune system. But now just one rule of thumb, viral infections cannot be treated by antibiotics because antibiotics only work against bacteria, not viruses. So now antiviral drugs can help treat viral infections, but not cure them. Of course, is there really a difference between treating an infection and curing an infection? But they inhibit the synthesis of the viral DNA by interfering with the assembly of the viruses. So emerging viruses, what we mean by emerging viruses are things that suddenly appear on the scene, suddenly become apparent. It could be something that was, in the environment or in wild animals or something for quite a period of time, but they're things that suddenly become noticed to people. For example, the Ebola virus, one of a lot of different emerging viruses that cause hemorrhagic fever, hemorrhaging and bleeding, in other words, and a lot of times a fatal illness. So Ebola virus in Africa, you may remember, the Ebola virus in Africa in 2014, 2015. You may remember the chikungunya virus from just a few years ago, and also the Zika virus that's transmitted by mosquitoes that became an emerging virus back in 2015, I think maybe 2016 as well. So there's quite a variety of things. So here's what Ebola virus looks like. A very nice color photo. And these were all taken under electron microscopes. Chikungunya virus, kind of a unique pattern there. And then the Zika virus, looks like a strange orb, or maybe looks kind of like the Death Star from Star Wars. I don't know. But the Zika virus. So in 2009, there was, if you may remember, 2009, there was an outbreak, a general outbreak in the general population. An epidemic usually is an outbreak of a large number of cases in a population. Flu-like illness that's a type of influenza virus called H1N1. And it caused, and Now, when there's a global epidemic, then it's called a pandemic, and we're very familiar with that term now. So pandemic means basically pan means widespread usually. So pandemic means an epidemic that's basically worldwide. And so there are three things that contribute to the emergence of new viral diseases then. RNA viruses have an unusually high rate of mutation, so they can change easily and elude the immune systems easily. It can be disseminated, that is spread from a small isolated human population. It can eventually spread around the world, like we saw with coronavirus. About 3/4 of new human diseases originate by spreading to humans from animals, actually, from one type of animal or another. So flu epidemics can be caused by type A influenza viruses, but they infect a wide variety of animals, including birds, pigs, horses, as well as humans. Different strains, different types of influenza A are given standardized names based on the viral surface proteins, or the hemagglutinin. Hemagglutinin, then a surface protein on a virus that causes the antibodies, human antibodies, to react against them. So the hemagglutinins, the HAs, or the neuraminidase, the NAs. And neuraminidase is actually a type of enzyme, and anything that ends in ASC generally is some sort of enzyme, just as a good rule of thumb. Like the reverse transcriptase that we looked at a few slides ago. So H1N1 then is the influenza A strain that caused the 2009 pandemic. So changes in host behavior or the environment can increase the spread of viruses of emerging diseases. So if they construct new roads into remote areas, that could increase the spread of viral diseases, because it could bring certain wild animals in contact with people at a higher rate than normal. Because if you're building new roads into a remote area, then you're bringing a lot more people into a remote area where there used to be very few. And so you're increasing the chance that they can come in contact with animals that may give them certain diseases. The use of insecticides and mosquito nets might help prevent the spread of viruses, especially viruses carried by mosquitoes, and also malaria, which is a protist carried by mosquitoes. So it's possible that maybe global climate change might allow mosquitoes that carry different viruses to expand their range. There are certain mosquitoes that are adapted to tropical climates or warm climates, and they cannot survive in warmer climates. But if the global climate gets warmer, then the area of warm, the areas of warmer weather, warmer climate in the world will expand, and that might expand the range of mosquitoes and other things that spread different types of disease. And here you can see, certain types of mosquitoes. Here's a type of mosquito that can transmit certain viruses, like Zika and other things. This mosquito is called Aedes aegypti, actually, Aedes aegypti. But if you have mosquito net to protect sleeping people, then they can be protected by the bite of mosquitoes, whether they're infected or not. What's a problem, though, is that in really, really warm tropical countries, sleeping in a mosquito net is kind of hot, because the mosquito net cuts down on the breezes blowing through. But anyway, just one of the challenges. So more than 2,000 types of viral diseases of plants are known and cause spots on leaves and fruits, or stunted growth, or damaged flowers or roots. But most plant viruses have an RNA genome. Many have a helical capsid, but others have an icosahedral capsid, like we saw in the very first few slides of this chapter. So like a damaged fruit, for example, here. Uh, and plant viruses spread diseases by two major routes, what's called horizontal transmission um, from one cell to another, or vertical transmission, inheriting the virus from a parent. So when they produce, when the plants produce offspring, they could actually pass on a virus as well. So prions then are infectious proteins, just proteins. They don't have nucleic acid with them. Infective proteins that appear to cause degenerative brain diseases in animals. Diseases called scrapie in sheep, mad cow disease in cows, you may have heard of, Creutzfeldt-Jakob disease in humans, caused by prions. And prions are basically incorrectly folded proteins. They can be transmitted in food, they can act slowly, and they're virtually indestructible. So prions are somehow able to convert a normal form of a protein into a misfolded version of the protein. And then several prions can aggregate or clump together into a complex that can convert more proteins to prions. And so these prions might be involved in diseases such as Alzheimer's disease and Parkinson's disease even. So here's you got normal proteins here, and here are the prions, the misshaped prions. So they attach to normal proteins and produce more. So here's the original protein. Here's the new prion. They convert existing proteins to misshapen prions, and pretty soon you've got aggregates of prions then as well. And so a lot of research is being done currently then on how to control prions then as well. So that's basically all of chapter 19 then. So look through it and study through it. And as always, if you have questions about chapter 19, contact me either through email or whatever, and I can answer your questions then. So anyway, keep studying then. And as always, remember that every day you should check your email and check your announcements in Canvas to keep up with all the latest events and all the latest things going on in this class and in other classes. So until next time, thank you, and have fun studying biology.